Hi everyone, and welcome to the MTOZ Biolabs channel. Today, I'll be introducing a classic and widely used quantitative proteomic strategy, SILAC. SILAC stands for Stable Isotope Labeling by Amino Acids in Cell Culture. As the name suggests, it's a quantitative proteomic strategy that incorporates isotope labels into proteins during cellular metabolism. In this video, we'll walk through the development, principles, advantages, limitations, and key applications of SILAC. The development of SILAC has been closely tied to its widespread use in small molecule quantification. Around the year 2000, Chait and Lom first explored metabolic labeling of microbial proteins, using carbon-13 and nitrogen-15 labeled culture media, enabling relative protein quantification. In 2002, Ong refined the amino acid-coded mass tagging approach originally proposed by Chin, leading to the formal establishment of the SILAC method. In recent years, the integration of SILAC with HPLC-MS has made it one of the most effective tools for quantitative proteomics. SILAC is now widely used in both functional protein studies and large-scale differential protein expression analysis. SILAC works by incorporating stable isotope-labeled amino acids into proteins through cellular metabolism. First, target cells are cultured in media containing either light or heavy labeled amino acids. The light media contains naturally occurring amino acids, such as carbon-12 or nitrogen-14-less or ARG, while the heavy media contains stable isotope-labeled amino acids, such as carbon-13 or nitrogen-15-less or ARG. Next, cells undergo at least 5 to 6 passages to ensure that the natural amino acids in the heavy media are fully replaced by the stable isotopes. Once stable isotope labeling is complete, the cells are subjected to experimental treatments, such as drug stimulation or gene knockout. The light and heavy labeled cells then represent different experimental groups. After treatment, proteins from each group are extracted, mixed in equal amounts, and then digested. Finally, the samples are analyzed by mass spectrometry. At the MS1 stage, the mass differences between labeled peptides are detected, and by comparing peak intensities, the relative abundance of proteins can be determined. SILAC offers several advantages. By incorporating labels during protein synthesis, it allows for standardized downstream processing and significantly reduces batch-to-batch variability, thereby achieving high quantification accuracy. Additionally, SILAC supports multiplexing, allowing the simultaneous labeling and quantification of multiple samples, greatly improving experimental efficiency. Furthermore, SILAC is flexible, as it is applicable to various cell lines and can be combined with other techniques, such as PTM analysis. However, there are some limitations. First, the process is time-consuming, as cells require 5 to 6 passages to ensure complete amino acid replacement, making the labeling process lengthy. Second, it is costly, as stable isotope-labeled amino acids are expensive, which may be a budgetary concern. Finally, SILAC is currently limited to conventional cultured cell lines and cannot be directly applied to tissue, body fluid, or clinical samples for quantitative analysis. SILAC is widely used for proteomic studies at the cellular level. It is commonly applied for differential protein expression analysis, such as comparing protein abundance changes between drug-treated and control groups. In this study, SILAC-based proteomics was used to assess how the humanized tumor-targeting antibody 14F7 affects protein expression in HeLa cells. Using light-labeled control and heavy-labeled 14F7HT treated cells, the team performed LC-MS to compare global protein expression. Over 3,600 proteins were identified, with 11 proteins significantly down-regulated, and 1 protein up-regulated in response to 14F7HT treatment. A volcano plot was used to visualize proteins with both strong fold changes and statistical significance. Further analysis, such as pathway enrichment, can reveal which biological pathways these differentially expressed proteins are involved in, providing insights into the cellular processes affected by 14F7HT. SILAC is also used in dynamic studies of post-translational modifications, helping to quantify changes in modified peptides with enrichment strategies. This case demonstrates how SILAC is used to study the dynamic changes of lysine acetylation and ubiquitination. The experiment integrates triple SILAC-based quantitative proteomics, with antibody-based enrichment techniques targeting acetylation and ubiquitination. The cells were treated with different compounds, DMSO, the ATN, a combination of aspirin, tricostidine A, and nicotinamide, and MG132, a proteasome inhibitor. Through LC-MS analysis, we quantified the protein modifications in response to these treatments. The results revealed that ATN combo treatment significantly increased acetylation levels without affecting ubiquitination. Conversely, MG132 treatment only influenced ubiquitination, but did not alter acetylation. This study highlights the power of SILAC in performing dynamic PTM analysis. Additionally, SILAC can be combined with co-IP for protein and protein interaction studies, effectively distinguishing specific binding proteins from background noise. Here, we present a case. The research aimed to identify the E3 ligase, responsible for the ubiquitination and degradation of the transcription factor E2F2. The research team established his E2F2 overexpressing HEK293T cells and used SILAC labeling for both the control and drug-treated groups. Co-IP was then performed, followed by LC-MS to quantitatively screen for interacting proteins. A total of 706 interacting proteins were identified, with 4 of them related to the ubiquitin proteasome pathway. Further focus was placed on 2 candidate E3 ligases, ZFP91 and TRIM25. Co-IP experiments revealed that the nuclear interaction between E2F2 and ZFP91 was significantly stronger than with TRIM25. siRNA-mediated silencing of ZFP91 notably reduced the ubiquitination level of E2F2 and inhibited its degradation. Ultimately, in vitro ubiquitination assays confirmed that ZFP91 is a functional E3 ligase. This case illustrates that combining SILAC with co-IP and mass spectrometry analysis is a powerful tool for accurately identifying specific binding proteins in complex backgrounds. At MTOZ Biolabs, we offer a comprehensive platform of quantitative proteomic solutions tailored to meet diverse research needs. Our SILAC-based quantification service enables interference-free, highly reproducible endogenous labeling by incorporating stable isotope-labeled amino acids during cell culture, ensuring accurate protein quantification. In addition to SILAC, we offer isobaric labeling strategies such as TMT and iTrack, which are ideal for high-throughput analysis of multiple samples, including tissues and biofluids. For precise quantification of target proteins, we offer absolute quantitative analysis services. It's especially suitable for target validation and clinical research. We also support label-free quantification for large-scale or discovery phase studies, as well as semi-quantitative approaches based on spectral counting or peak intensity. These options are cost-effective for exploratory or budget-constrained projects. Whether you're working with cells, tissues, or biological fluids, we can help you design the optimal quantification strategy to ensure efficient experiments and reliable data. Thank you for watching and feel free to contact us for more information.